with that, we have a very interesting lecture today, and so you need to be on top of it because we will go speed up a little bit. We've been very relaxed, and we will continue being relaxed and enjoying, but we'll speed up a little bit. So we talk about how the innate immune system can have different phases, preventing infection, and we were talking about recognizing the microbes in previous lectures, and we talk extensively about extracellular proteins or soluble proteins that recognize the microbes, like the complement system, that is a group of protein that can recognize the microbe or be activated upon recognition of the microbe, and then can lead to alarm and destruction of the microbe. And today we will cover cell-associated receptors that will also recognize the microbe. And we already talked about what are pathogen recognition receptors, which are receptors for pumps in the pathogens, and how they will always signal through an adapter and a cascade of kinases to initiate transcription that will alarm and destroy. And we will look more into detail about that today, and then we will try to get to how alarming occurs and how destruction occurs. Okay, so just an overview in this slide for the different cell associated receptors that will detect the presence of a foreign substance. We have receptors that are associated to the membrane, as we mentioned, that will localize in the plasma membrane. And examples for that are toll-like receptors that we will focus on, mannose receptors, scavenger receptors, and so they are all localized in the plasma membrane. And we also have membrane-associated receptors that are associated with the endosomal membrane, and the pathogen binding domain will be towards the internal endosome. And so we will focus on some of the toll-like receptors that are associated with the endosomal membrane as well. And then we have pathogen recognition receptors that are not associated with membranes, and that includes node-like receptors and also the CARD family of sensors, as well as a more recently described a receptor that is called cGAS. And it's not a receptor, it's an enzyme, and we'll look into that as well. So there are small changes that I made last minute in the slides to update, to make it more organized, and I will upload the updated PDF right after the lecture. Okay, so let's focus on the membrane-associated receptors. So we have we will use as example the toll-like receptors, and so these toll-like receptors are a type of pathogen recognition receptors. And as you know already, the pathogen recognition receptors recognize molecules that are broadly shared by pathogens, but are distinguishable from host molecules. And these molecules or patterns that again are shared by a group of pathogens, but are distinguishable from host molecules, they are called pathogen-associated molecular patterns and PAMPs, as we mentioned in the previous lectures. So these toll-like receptors receive their names for their similarity to the protein coded by the toll gene, that is a gene that was identified first in Drosophila by Hoffman, and that earned him the Nobel Prize. And later, Euler identified the first ligand of the mammalian toll-like receptors being a LPS for toll-like receptor 4, as we will see, and he shared the Nobel Prize in 2011 with Hoffman. And so interesting, when Drosophila, Hoffman found that when Drosophila lacks the gene toll, they become more susceptible to bacterial and fungal infection. And that was the first evidence that this toll may participate in host defense. Okay, so where do these toll-like receptors localize? We have some that will localize associated with plasma membrane, and that includes toll-like receptors 4 that which pump or which ligand in the pathogen is the lipopolysaccharide or LPS. Toll-like receptor 5 also localizes in the plasma membrane and recognizes gelin, which is present in some bacteria and is used for bacterial locomotion. Toll-like receptor 1, 2, and 6, as well as toll-like receptor 6, also localized in the plasma membrane, and they recognize lipopeptides from microbes. And then we have 3 toll-like receptors that localize in the endosomes that are toll-like receptor 3 that recognize double-stranded RNA. I think we mentioned in one of the previous lectures that this double-stranded RNA is produced in any viral life cycle. So it makes sense that we have a receptor that will recognize this pump, right? Pathogen-associated molecular pattern, the double-stranded RNA, because produced in any viral life cycle and is not produced in our cells. The receptor 7 is also localized in the endosomes, and it senses the presence of single-stranded RNA. And this is important for many RNA viruses that are single-stranded RNA, like, for example, influenza virus, SARS coronaviruses, HIV, are all single-stranded RNA viruses, and they are all sensed in part via toll-like receptor 7. And then we have also in the endosome toll-like receptor 9, which a pump ligand is CpG DNA, particularly unmethylated CpG DNA that is abundant in the DNA of bacteria and viruses, like, for example, herpes virus or, for example, Mycobacterium tuberculosis, but not in our own DNA. On top of that, you need to remember, and we'll try to point this a concept later as well, is that our self-DNA may have some unmethylated CpG, but that will not be present in the endosome. So the pathogen recognition receptors can distinguish the pumps in the pathogen, not only because the structures that they recognize and that they bind are more abundant in the pathogen, but also because of the localization of these structures in the context of an infection versus the localization of self-DNA, for example, that will never be in the endosome, okay? Okay, so we mentioned that LPS is a pump, is actually the first pump that Bruce Boiler identified as the ligand for the first mammalian toll-like receptor, and this is LPS being the for toll-like receptor 4. And LPS is the component of the cell wall of gram-negative bacteria like Salmonella or Escherichia coli, and basically is constituted by a long saccharide chain together bound to a lipid, okay? And this is what toll-like receptor 4 will recognize. LPS is also known as endotoxin, and the reason is because the presence of LPS in circulation, or this endotoxin in circulation, leads in blood circulation, leads to systemic inflammation that can cause septic shock. And we will learn more about that with a dedicated slide for septic shock. So one interesting concept is that bacteria and other microorganisms may be recognized by more than one path recognition receptor. And that makes sense because we want to maximize the chances that we recognize the presence of these microbes, so we can then trigger an alarm, and also pathways that will, or mechanisms that will destroy the pathogen, right? So the more PRRs that recognize the microbe, the better. And an example is here, so some bacteria, some gram-negative bacteria can be recognized by toll-like receptor 4 binding to the LPS in the cell wall of the gram-negative bacteria, but some of them have also a flagella component, and that can be recognized by receptor 5. And so again, more than one pathogen recognition receptor can be used to recognize the same pathogen. One additional piece of information is, and that's particular for toll-like receptor 4, is that it not only requires toll-like receptor 4 binding to LPS, but it also requires other proteins in this complex, particularly toll-like CD14, which should also bind to another part of the LPS molecule. And so LPS recognition requires not only toll-like receptor 4, but also the recognition of LPS via 14 molecule, which is another receptor that is expressed, for example, in macrophages and other innate cells. And this brings me to the definition of what is a CD. So a CD, and because you will be hearing throughout this course a lot about this CD, and this case is CD14, but we will, there are many other CDs, and basically this is a CD means cluster of designation, and it's a protocol used for the identification and investigation of cell surface molecules that then provide targets for immunophenotyping of cells, okay? So many times we identify the cells of immune system but, because they express a combination of these CDs that allow us to identify the immune cells or immunophenotype the immune cells. So we know if it is a macrophages, a neutrophils, a B cell, for example, and you will see how this is used when we learn in discussion section, special topics, flow cytometry. Physiologically, the role of the molecules varies, and they can be either receptors or ligands, but we use them to identify immune cells. Okay, so we again mention that these pathogen recognition receptors will always work in association with adaptor proteins that are intracellular and will trigger, will mediate and enable the signal events after the PRR have been engaged by a pump, right? And so for the toll-like receptors depicted here, the majority of them act via the adaptor MYD88. And this will initiate the activation of a signaling pathway that involves, most of the time, the signaling downstream of the pathogen recognition receptors involve kinases. It's a cascade of kinases most of the time. And in this case, downstream MYD, toll-like receptors and MYD-ADI, there will be the activation of the kinase, many kinases, including AKK kinases. And these kinases then ultimately will activate transcription factors. And so particularly AKK will phosphorylate the inhibitor of NF-kappa beta. And by phosphorylating this inhibitor, it will promote the degradation of this inhibitor. And so now NF-kappa beta will be free to translocate into the nucleus and promote the transcription of cytokines and chemokines, which are the products that will lead to alarming and destruction of the pathogen. So when toll-like receptor 4, for example, senses the presence of a gram-negative bacteria because it is engaged by LPS, MYD-ADI will trigger a signaling that to AKK activation, NF-kappa beta translocation into the nucleus, and activation of cytokines and chemokines. And that's fantastic because these cytokine and chemokines now will go and alarm the body that there is a presence of a gram-negative bacteria and trigger mechanisms for its destruction. Some of the toll-like receptors can also use, as we will see in the next few slides, another adapter that is called TRIF. Okay, so I will review that in the next couple of slides. So here you can see again, this is what I show you already, that TLR4 and other toll-like receptors will lead to the, when engaged by the bacterial or other microbe pumps, will lead to MYD-ADI and AKK kinase cascade with the activation of AKK kinase and the activation of NF-kappa beta via the degradation of the inhibitor of NF-kappa beta. And now NF-kappa beta can translocate into the nucleus and initiate the production of cytokines and chemokines. As I said, some of the toll-like receptors, including the toll-like receptor 4, can also operate via the, in addition to use MYD-ADI an adapter to trigger NF-kappa beta and produce inflammatory cytokines, and we will see these are some of them, IL-1, IL-6, TNF-alpha. They also use as an adapter TRIF, okay? So when they are engaged by LPS, not only will lead to the activation of MYD-ADI and NF-kappa beta activation, but will also lead to the activation of TRIF adapter in an MYD-ADI independent pathway, and this will lead to the activation, phosphorylation via the kinase signaling cascade, phosphorylation of IRF3. And in this case, the kinase that is involved is TBK1. And this IRF3 then is a transcription factor that will, when phosphorylated by this TBK1 kinase, can translocate into the nucleus, but now will bind to the promoters of genes that encodes type 1 interferons. And these type 1 interferons are a group of proteins that include interferons alpha and interferon beta. Okay, there are about 13 interferon alpha isoforms and interferon beta, and we will see in the next slide what do they do. But they are important also for alarming and destruction of the pathogen. And so just a piece of vocabulary here, IRF stands for interferon regulatory factors, and these IRF are transcription factors that are involved in the transcription of interferons, okay? Okay. So this is I would like to cover for the membrane-associated receptors using toll-like receptors as the main example. We also have cytosolic receptors, as we just mentioned, and that involves the node-like receptors. Again, these are in the cytosols. They can also sense a bacterial infection now when it is present in the cytosol, and they will also lead to NF-kappa beta activation. And so, for example, if you have a bacteria, particularly protoglycan of a bacteria, that can be recognized in the surface by the toll-like receptor 2, but these bacteria may also have muramyl dipeptides, and this is a pump that is present in gram-negative and gram-positive bacteria, and this will be recognized if the bacteria is invasive and reach the cytosol of the cell. Now it will be recognized by these node-like receptors and induce the activation of NF-kappa beta. And you know that NF-kappa beta will translocate into the nucleus and promote cytokine and the expression of pro-inflammatory cytokines like IL-1, IL-6, and TNF-alpha, and we will learn more on how they act. Okay, so again, another family of cytosolic receptors, apart from the node-like receptors, are the card-like receptors. And these card-like receptors, card family sensors that include RIG, for example, they recognize cytoplasmic double-stranded RNA. And again, here we have another example on how the same pump can be recognized more than one pathogen recognition receptor. For example, double-stranded RNA, we mentioned in the previous slide, can bind to toll-like receptor 3 in the endosome. And in this case, when it binds, it can signal via toll-like receptor 3 uses TRIF as an adapter, and this will lead to the activation of IRF proteins. So TRIF always leads to the activation of IRF proteins and the production of interferons. Remember, IRF proteins are transcription factors that induce transcription of interferons, okay? But double-stranded RNA can also be sensed if it is present in the cytosol by RIG-like receptors, RIG-I, which is this card family sensor, belong to these card family sensors. And again, that will induce the activation of IRF proteins that will lead to the production of interferons. So this is one way in which signal the presence of viral RNAs, and also, to be honest, this double-stranded RNA can be produced as an intermediate in DNA viral infections, right? But we mentioned for other toll-like receptors, like toll-like receptor 7, being important to recognize single-stranded RNA, like is present in the genome of influenza, HIV, coronaviruses, right? But what about DNA? How is the presence of viral DNAs, particularly, or viral or bacterial DNA, particularly in the cytosol, being recognized? And so I switched here the slide because I thought that this would be much clearer for this course. And so here you can see depicted how DNA from viruses or bacteria, when they reach the cytosol of the cell, they can be recognized by this enzyme that is called SIGAS. It has only been identified a few years ago, and it's called cyclic GMP AMP synthesase, SIGAS. And so this enzyme will bind cytoplasmic DNA. So we shouldn't have DNA in our cytosol. Our own DNA is in the nucleus. So if we have DNA in the cytosol, it's that is sensed as a potential dangerous presence and foreign presence. And so this, once DNA is bound by this enzyme, the enzyme will convert DNA into a CGAMP intermediates, okay? So cyclid guanosine or adenosine monophosphate intermediates. And now these intermediates can activate an adapter that I'm not asking you to remember the name, and this adapter will lead to the activation of the same kinases we've been talking about, the activation of the AKK kinase that will lead to NF-kappa beta activation via degradation of the inhibitor of NF-kappa beta, and then translocation of NF-kappa beta to the nucleus, and production of pro-inflammatory cytokines such as IL-1, IL-6, TNF-alpha. And also this adapter leads to the activation of another cytokine that we mentioned, that is TBK1. And this TBK1, sorry, I want to say another kinase that we mentioned, that is TBK1. And this TBK1 will promote phosphorylation of IRF protein. And these IRF, when they are phosphorylated, we said they are transcription factors that now, when phosphorylated, can translocate into the nucleus and promote the transcription of interferons, okay? And so the pro-inflammatory cytokines and the interferons that results from this signaling will now lead to the alarming that there possibly there is a dangerous presence and the destruction of this virus or bacteria that are invading. And so, like always, the pump, in this case, the pump is the cytoplasmic DNA derived from a virus of a bacteria, will be recognized by a path recognition receptors, in this case is the SIGAS enzyme, and via an adapter will lead to a signal, most cases is a signal via kinases, and the activation of transcription factors that will lead to the production of inflammatory cytokines and interferons. Okay. So there is a nice global immunotalk that I'm putting the information here, delivered by Dr. Kate Fitzgerald from UMass, that it talks about recognition of this cytoplasmic DNA and how it is being used for therapies. So they are generating a agonist of SIGAS to promote production of cytokines and particularly interferons in the context of tumors, to activate inflammation in the tumor and enhance the responsiveness to point therapies and other therapies, but to help the host identify the tumor as a foreign presence. And so if you are interested in that, you can check that in Global Immunotalk. And again, it's very advanced, so feel free to disregard some parts, or you can do it after completing this course. Okay, so I guess here is the same slide. Again, we learned that not only toll-like receptors, but the RIG, the card sensors, and also the node-like receptors and the SIGAS, they are all pathogen recognition receptors. They signal via, always associated with some adapters. We talk about MYD88 and TRIF, and lead to a protein kinase cascade. We talk about AKK, leading to activation of NF-kappa beta transcription factor, and TBK1 leading to activation of IRF transcription factors that in the case of NF-kappa beta will induce production of pro-inflammatory cytokines like IL-1, IL-6, TNF-alpha, and IRF when translocate will promote transcription of type 1 interferons. And so this is another slide, just putting together the same and adding a little piece of information. Here again, we are depicting all the pathogen receptors, the PRRs, the toll-like receptors, the RIG-like receptors, also known as CARD family sensors, the DNA sensors like SIGAs. They will all act through different adapters, and we learned these two, MYD88 and TRIF, and that are used for by some toll-like receptors, and they will lead to the activation of BI kinase cascades that includes AKK and TBK1 kinases, to the activation of NF-kappa beta and IRF transcription factors. And so this will again lead to the activation of cytokines like TNIL6, and these are produced already in an active form. But there is an interesting fact that is that the transcription of IL-1 gene will produce a pro-IL-1 protein, which is inactive, okay? And so you will produce downstream this cascade an inactive form of IL-1 that is called pro-IL-1, okay? And so this pro-IL-1 to become activated requires the activation of caspase-1, which is a protease that would cleave the pro-IL-1 to produce the active form of the IL-1. And how is this caspase-1 activated? Well, many of these node-like receptors that are part of the inflammasome system, is called also inflammasome system, can lead to the activation of caspase-1, okay? And again, this is important because this caspase-1 is the only one that can cleave pro-IL-1 beta into active IL-1 beta, and this IL-1 beta not only exert inflammatory properties, but many times can induce cell death. The caspase-1 activation can many times lead also to cell death and death of the infected cell. Okay. Okay, let me see. So we will do this, I'll try to do this poll question now, if possible. Let's see. But before I do that, I want to summarize a little bit, right, here, and just give you a perspective on where we are here. So basically, talking about how the innate immune system can prevent the entry of a pathogen, can recognize, and we have been looking at how it can recognize the presence of the microbe. And we talked past lecture on how we can recognize it with a complement system, and that is a soluble group of proteins. And today we focus on how we can recognize it by cellular associated receptors that can be in the plasma membrane or endosome, but also membrane associated receptors, or they can be in the cytosol, okay? All these receptors will always recognize pathogen products, pathogen patterns, like we talk about toll-like receptor 4 recognizing LPS, or we talk about how RIG-like receptors or the node-like receptors recognize double-stranded RNA and muramyl dipeptide in the cytosol, respectively, okay? And so they always bind PAMPs, pathogen products that can be distinguished from host self molecules. And this leads, it will activate another that is different from different pathogen recognition receptors, and we talk about MYD88 and TRIF being adapters for some toll-like receptors. And after that, there is a signaling cascade with kinases, and we talk about AKK, and we talk about TBK1 kinase, which will activate transcription factors. So AKK will activate NF-kappa beta, TBK1 will activate IRF transcription factors. And these transcription factors will then promote the transcription of cytokines and chemokines that will alarm and destroy. The NF-kappa beta will promote activation of what we call pro-inflammatory cytokines and chemokines, including IL-1, IL-6, TNF-alpha, and the IRF will promote the transcription of interferons that we will see in the next slides how they act. Okay, so any question about that? Okay, so let's do this, the first one together. So here, this refers to a graph that I think that we put in the first lecture. And basically, this is here the duration of infection and the number of microorganisms. And so if you remember in the first lecture, we said if you have innate and adaptive immunity in the best case scenario, you will be able to control a pathogen and initially delay its replication, and then the adaptive immune system will completely clear the pathogen. Now, if you lack the adaptive immune response, this pathogen will be be controlled by the innate system initially, and then will lead to but will not be able to eliminate because you lacked adaptive immune response. This would be the case in a T-cell deficient host. So in this case is a viral infection with single-stranded RNA virus, could be coronavirus here. And so if you don't have T cells, then you will never be able to control the virus, but you may be able to delay its replication by the innate immune system. So now if you lack components of the innate immune system that are important to sense single-stranded RNA virus, then you will have a rapid growth and spread of the virus, okay? So the number of microorganisms in the infected host will grow significantly. And so the normal host, again, this case will be C. And let's see, I will ask you to do these questions. So I will put the poll here and you need to pick which option will be the most appropriate. And again, it says the graph on the left depict the amount of single-stranded RNA virus after infection of different hosts. Indicate which graph, A, B, or C, most closely represents the following host. And I'm asking you to answer the question for this host that is a host that is deficient in MYD88, is deficient in IRF3, and is deficient in IRF7. And so again, this is a single-stranded RNA virus. And okay. So let's see what you guys answer. I will. Do you see the poll?
Yes.
Okay, perfect. Thank you. Okay, so let's stop end the poll now and let's discuss. Okay, I will end it now. And so share results. And so most of you answer A, and I would agree with that because a single-stranded RNA virus will be recognized by toll-like receptor 7, right? That's what we said. And toll-like receptor 7 signals via MYD88. That's the adapter of one of the adapters, and that will lead to NF-kappa beta activation. And so you will not have activation of NF-kappa beta because you don't have the adapter in the first place. In addition, like any virus, this virus will produce in the cytosol double-stranded RNA, right? When it replicates inside the cell, right? The viruses don't replicate in the endosome, they replicate inside the cell and in the cytosol or the nucleus. And so they will have a generation, this virus will generate double-stranded RNA as an intermediate. And this double-stranded RNA is sensed by these card-like sensors or RIG-I. And, but remember, to produce any interferon, you need IRF3 or IRF7, okay? So you will not activate NF-kappa beta, and you will not activate IRF, or you will not be able to activate interferons because they are not, there is no IRF. So there could be some other activation of the innate system, but the major pathways in which this single-stranded RNA will be sensed by the immune system will be massively suppressed. And so I expect that it will be the most closely representing the number of microorganisms in this host that lacks MYD88, IRF3, and IRF7. Okay, we will not do these ones, and I will leave them for the discussion sections. They are also interesting to think about, but we will leave these for discussion sections. Okay, so let's move to the other phases of the innate immune response. So once the pathogen is recognized, and we said there will be signaling and transcription of cytokines and chemokines, and including interferons. And so this will alarm other cells and tissues. And so interferons are cytokines as well. And so what are exactly cytokines? Cytokines the word derives from the Greek cyto, that means cell, and the means movement, and are small cell signaling proteins or molecules that are secreted by numerous cells to affect the behavior of other cells. So they participate in cell-to-cell communication, basically. And just as a matter of vocabulary, I need to tell you what is interleukins. So interleukins, many times I use like synonymous as cytokines, and is a group within these cytokines that were first discovered to be expressed in leukocytes. That's why they are called interleukins. The term interleukin derived from inter, because as being a cytokine are important for cell-to-cell communication. And last, so basically these are synonymous, basically. You can use them as synonymous. And last is chemokine, is another vocabulary. And again, is kainos is referred in Greek to movement. And these are a family of small cytokines with the ability to induce directed chemotaxis in nearby responsive cells. And so they are chemotactic cytokines, basically direct the movement of cells that are responsive and have the receptors for chemokines, okay? So all cytokines and chemokines, including the chemokines, they always will act in the cells that carry the receptors for these cytokines or chemokines. And we will see what is the consequence of the cytokine binding to the receptor. Is different for different cytokines. And so this, just to put again in perspective here, we talk about how macrophages were phagocytic cells. And so when a bacteria binds to a receptor in a macrophage or other innate cells, that is triggering this receptor may trigger the engulfment of the bacteria, and this will promote the degradation of this bacteria. But it's possible, and most likely, if it is a macrophage that like we talk about professional innate cells that have many pathogen recognition receptors, that the bacteria will also engage a PRR, a pathogen recognition receptor. And that, as you know, will induce transcription of cytokines and chemokines that not will be released by this macrophage to alarm and destroy. Okay, just to put in context a little bit what we talk about, who will be producing these cytokines and chemokines. So macrophages and other innate cells can produce cytokines and chemokines. And honestly, some chemokines are produced by in endothelial cells, or epithelial cells can also produce chemokines. Okay, how these cytokines and chemokines will alarm. And so we will cover 4 ways in which the cytokines and chemokines can alarm that there is a potential danger. One is by the inducing this mannose binding protein of C-reactive protein, and we'll look a little bit more in the next slide how they can opsonize pathogen and activate complement. But number 1 is the production of these 2 proteins. Number 2 is the activation of other cells. Okay, the cytokines can activate other cells. Number 3 is recruiting other cells. We talk about how chemokines can attract and modulate or direct the movement of other cells, so they may recruit other cells. That's particularly for the chemokines. And 4, these cytokines can have systemic effects. And so we will go through these 4 mechanisms via which the cytokine and chemokines can alarm our body that there may be a dangerous presence, okay? Okay, so the first one we said, the cytokines may induce the production of mannose binding lectin or mannose binding protein and the C-reactive proteins. And you may remember, and I know that we mentioned many new names, but you may remember that we mentioned these proteins already. Any brave one that wanna suggest where did we mention these proteins?
Isn't it one of the pathways, one of the 3, the second pathway of the innate immune response?
Yes, you are super close. Thank you so much, Sydney. So we'll see. So these cytokines, for example, one of the pro-inflammatory cytokines we just mentioned in the previous slides, interleukin-6, is able to signal in the liver for the hepatocyte to produce C-reactive protein and mannose binding lectin. And so the C-reacting protein, as well as the mannose binding lectin, will bind to the pathogen and will opsonize the pathogen. What does it mean, opsonization? 5 points for the one that, no, it's a joke. But yeah, who wants to answer?
Isn't it any molecule that enhances phagocytosis?
Excellent. That's opsonization. It's that simple. Enhances the phagocytosis. So now, by being bound by the C-reactive protein of the mannose binding lectin, now these bacteria here will have more chances and be more effectively recognized and uptaken by a macrophage, will be opsonized, okay? And also these 2 proteins, as Sydney was referring to, we mentioned them because they were able to initiate the activation of complement. So the pathway is initiated by mannose binding lectins that bind to the surface of the pathogen, right? So we mentioned them before and can initiate complement activation. And also the C-reactive protein can initiate this classical pathway of complement activation. And so both of them will lead to further activation of the complement after they are produced by the liver. And you know, that's why they are not as quick as the alternative pathway, which doesn't require any protein to be produced by the host. It just requires the complement to bind to the surface of the pathogen, okay? That's why the lectin is second to act, because it requires the mannose binding lectin to be produced. Okay. So that's one of the mechanisms via which cytokines can alarm, right? By inducing the production of this mannose binding lectin and C-reactive lectin. And there is another mechanism how the cytokines can alarm, and is by inducing the activation of other immune cells. And an example for that is the production of interferons. And we mentioned that type 1 interferons include interferon alpha and interferon beta, and which are the factors that promote the transcription of type 1 interferons. Any brave one? Which transcription factors will induce the synthesis, the transcription of genes encoding for type 1 interferons?
Isn't it IF3 and IF7?
Yeah, IRF3 and 7. You can, if you remember IRF, that's, I'm happy enough. Yeah, thank you so much. So these, but what happens when they are produced? They can signal via their receptor, and they will induce an antiviral state in a paracrine or autocrine manner. And we will see how they induce an antiviral state. And they can also activate natural killer cells by their killing capacity, or by enhancing the expression of activating receptor natural killer cells. And we will learn more about natural killer cells later, okay? But remember that interferons can activate these natural killer cells. Okay, so how interferons induce this antiviral state? And this is very, very timely now, because I show you in the first lectures how there has been shown that there is a concentration in the people that suffer from severe COVID-19 that have inborn mutations in genes that encodes for the interferon system, including mutation interferon receptor 1, but also mutation in IRFs, okay? And what it has been shown is that many of the patients that develop a severe COVID-19 have mutations in these important genes and factors that we are learning in the lecture today. And so we learned already how interferons are induced. So we will not repeat that, but remember these are pathogen recognition receptors that will lead to the activation of the IRF. IRF will promote production of interferon alpha and beta. And now this interferon is secreted, right? Like all cytokines are secreted. And they will act in the interferon receptor, okay? And I will not ask you to remember, but just in case if you want, this will induce signaling via STAT proteins, And these proteins are transcription factors too, so they will translocate into the nucleus and will promote the transcription of hundreds of genes. So now interesting, this transcription is not just in the cells that have seen the pathogen, but this interferon may go to another neighboring cells that never saw the pathogen, but all the cells in our body express interferon receptors. And so the interferon may be alerting a cell that still is not infected with the pathogen, which is fantastic because now it's giving that cell the heads up, right? And so now any cell where interferon is binding to interferon receptors, there will be production of these interferon stimulated genes, which are hundreds of genes, okay? So we'll not go through all of them. But just some example, some of them are these RIG receptors, right? The card family of sensors. So it will increase the expression, further increase the expression of these receptors. So the neighboring cell, for example, that hasn't been infected, can now be more ready to sense the presence of a virus because it will express very high levels of these pathogen recognition receptors. In addition to that, it will induce some sensors of double-stranded RNA, which are not normally expressed in any cell under constitutive steady state conditions. But when interferons are present, they will induce high levels of these additional sensors of double-stranded RNA, which will lead to the degradation of RNA from the virus, as well as inhibition of translation. And that, of course, will minimize or counteract the ability of the virus to produce new particles. And so this is just two examples. And again, there are hundreds of genes and they all have different capacity to create an antiviral state in the cells where interferon signal. It can be in a paracrine manner, so it can be, in an autocrine manner, same cell that produce the interferon, or they may signal in a paracrine manner, in a cell that is neighboring, that is not the one producing the interferons. And so if you didn't believe me, this is a paper here that shows in a mice that are deficient in interferon receptors here with the black square, these mice are way more susceptible to coronavirus infection. And so you can see here is the survival of a wild-type mouse infected with mouse coronavirus, and this is here the death, rapid death of the interferon receptor deficient mice, and you can see that the virus killed the mice because it spread much easily, not only in the lungs, but also in the brain, for example. So type 1 interferons are critical antiviral mediators. And as I mentioned, all cells have, for example, expression of this card family of sensors. And so all cells, including the cells in our lung epithelium, have these sensors. And so it have the capacity to sense the presence of the virus when they are infected, and they can produce interferons. And so I think that this is depicted here. What happens is that many viruses have evolved means to block the induction of interferon in the cells where they replicate, right? Because that would be beneficial for the virus. And actually, SARS coronavirus 2 has very, very efficient means to inhibit the production of type 1 interferon in the cells where they infect. However, there are other cells that can produce interferons even if they don't have intrinsic viral replication. And they can produce 100 to 1,000 more interferons than any other cells. And these are plasmacytoid dendritic cells. And because they can produce interferon without being infected, basically they sense, they have a to sense neighboring infecting cells, they are not susceptible to these antagonistic strategies by the virus. And so they produce tons of interferons. And again, this interferon can act in a paracrine or a manner to protect infected cells and cells that are not yet being infected. And of course, viruses have evolved also, and depending on the virus, they are more efficient or not to suppress also interferon signaling. It seems that SARS coronavirus 2 seems to be most effective at suppressing the induction of interferon, but less effective at suppressing signaling of interferon. That's why there is a lot of hope with therapies that enhance or deliver recombinant interferon, because the signaling seems to be mostly intact. And so that's what we seem to know so far. Okay, so then we said the cytokines can alarm by inducing the production of manus-binding lectin and C-reacting protein. by activating other immune cells like the interferons do, creating an antiviral state or activating natural killer cells. And third, the chemokines, we said how they will recruit more immune cells. And I will not go through the details here to avoid adding more names, but basically the chemokines can be produced by endothelial cells and epithelial cells, and they will form a gradient that will attract the leukocytes that have the receptors for these chemokines. So, for example, a neutrophil or a macrophage will extravasate and will go to the site of infection where chemokines are being produced. And we will see more in one lecture all the molecules that participate in this leukocyte extravasation. And finally, the other way in which cytokines can alarm and help the destruction are via the induction of systemic inflammatory responses. And these are IL-1, IL-6, and TNF-alpha, which we mentioned before, are producing, are also called pro-inflammatory cytokines, and they have systemic effects. We talk about how the IL-6 can act in the liver, right, producing the C-reactive protein and the mannose-binding lectin. They can also act in the bone marrow, inducing the release of neutrophils that now will be more available in circulation to go to the site of infection, extravasate, and destroy the pathogen via phagocytosis. Particularly IL-1 beta can signal at the central nervous system, particularly at the hypothalamus, to which is can regulate the body temperature and basically increase the body temperature. And some of these cytokines can also act at the level of the fat muscles, promoting the mobilization of fuel, right, lipids and proteins that will be used to increase this body temperature. And this will induce fever, that is a typical response to an infection, but is induced by these cytokines acting at the level of the hypothalamus. And this will decrease viral and bacterial replication because they have evolved to optimally replicate at 37 degrees, not at a fever temperature. Okay, so we have learned in the previous lecture about inflammation when we talk about C3A and C5a increasing vascular permeability. So TNF-alpha can do the same, can increase vascular permeability. And so what I want to bring up today is that too much systemic innate response, for example, too much TNF-alpha may result in death, in septic shock. And so you may have a TNF-alpha inducing a local inflammation that will contain the infection, and that's good. But TNF-alpha is the systemic circulation may induce enhanced vascular permeability all throughout the body. So not just locally, but now is in the blood, and so it can affect all the the vasculature, and so that will cause a plasma to get away the vessels all throughout our body, and that will cause edema in our tissues, and will also decrease the amount of blood in our body and also the amount of protein in our blood, so creating hypoproteinemia. It will also induce a great expansion, a great depletion in the number of neutrophils that we have in the blood. That's another characteristic because they will all extravasate. And also, I didn't mention last lecture, but another way in which we can prevent a pathogen getting from the into the blood is because TNF-alpha can promote the production of platelet activating factors. And these platelet activating factors will promote the aggregation of platelets and coagulation, typically in local vessel restricted to the site of inflammation. And this will make the blood thicker, right? And will prevent the pathogen entering into the blood. This is one other mechanism. However, if this is occurring systemically, this will promote intravascular coagulation throughout the body, and all of these low blood pressure, because and also intravascular coagulation will lead to a septic shock with systemic edema and excessive coagulation that will starve all our organs and basically lead to death, okay? And so the septic shock is a consequence of too much inflammation or systemic inflammation, is when these pro-inflammatory products like TNF-alpha reach the circulation in very high levels. Okay, we will skip this. I think that this is also an interesting question for discussion section, and if you have more questions afterwards, I'm happy to answer in office hours, but I will include in the discussion section for this Friday and the following week. And so after the system, the cells are alarmed, the cytokines alarm other tissues, and remember we have 4 mechanisms that we checked on how they do it. There will be destruction of the microbe or infected cells by this microbe. And there are 2 mechanisms that I want to cover today for that. One is via phagocytosis, and the other one by cytotoxicity. Mediated by innate immune cells. So the phagocytosis we already talked about is mediated by neutrophils and macrophages, which are professional phagocytic cells. And so just as a review here, the neutrophils have 2 types of granules, that are the azurophic granules and the specific granules. And so when the neutrophils acquire or uptake a bacteria, maybe this bacteria is opsonized by the manus binding lectin, right? And so it will be uptaken even better by a neutrophil. So when this is uptaken, now this endosome or phagosome will be fused with these granules that are present in the cytosol of the neutrophil. And these granules have some antimicrobial products that will initiate the destruction of the bacteria, and they are characteristic by doing so at a high pH. Later on, the phagosome will bind with the lysosome. And as I mentioned when we were talking about macrophages, the lysosomes are also in the macrophages, and they have low pH. And this fusion of the phagosome with the lysosome will form the phagolysosome that will completely destroy the bacteria. And what happened with the neutrophil is that after it destroys the bacteria, normally the neutrophil is very short-lived and it will die. And like any apoptotic cells, and we will learn more about that, will be uptaken by a macrophage to clean up, right, like the debris. Okay, last mechanism for destruction is the cytotoxicity. And natural killer cells, as the name implies, they are very efficient at killing infected cells and also tumor cells. So the natural killer cells are always ready to kill, but as we mentioned before, their effector function can increase 20 to 100 folds when they are stimulated by cytokines like the type 1 interferons that we mentioned that can activate natural killer cells. And these cytokines are produced by other innate cells like macrophages or dendritic cell, for example. We mentioned how plasmacytoid dendritic cells are the ones that are most powerful producing produce 100 to 1,000 times more interferon than other cells. So this is here, this would be a natural killer cell that is ready to kill before interferon, but this is after they receive interferon. They are much more efficient at killing. And so how do NK cells recognize a cell that is infected so they know that they can kill that cell, right? We don't want NK cells to be killing any cell in our body, right? Because that will be unnecessary damage. And so they have two types of receptors. One inhibitor, the inhibitory receptors that tell them not to kill a target cells. And as an example of that is this inhibitory receptor that recognize molecules that are present in all nucleated cells that are called MHC class 1 molecules. And now it doesn't make any sense, this name, but when we study adaptive immunity, these are essential molecules for T cells to recognize antigens. So just hold on this name, okay? So the inhibitory receptors in NK cells will bind to MHC class 1 molecules that are present in all nucleated cells, okay? And so this will send a negative signal that will suppress the signal, the positive signal, typically induced by the activating receptor that binds a ligand. And ligands that are expressed in cells when they are stressed. For example, they may be stressed because they have a viral infection, and so they will express the ligand, so which is a flag that is like, potential danger inside. They also, the tumor cells, they are stressed cells, and they can express this ligand, and that's why NK cells can kill tumor cells as well. If the inhibitory receptor is not engaged, so if the cell is not expressed in MHC class 1, and many viruses suppress the expression of MHC class 1 to evade the recognition by T cells, and we will see why in the next few lectures. But when MHC class 1 is not expressed, now the inhibitory receptors cannot signal, and now you only have the positive signal by the activating receptor. And now the natural killer cells are able to kill this virally infected cell or this tumor cell, okay? Okay, any question about that? Okay, we'll do the summary in the next slide, and I'm happy to answer questions if you have. So what I wanna end up here is with the concept that different immune cells deal with different types of pathogens. And so if the pathogen is extracellular, now it can be localized in the interstitial space in between cells, in the blood, in the lymph, and in that case, it will be recognized by complements that we said, remember we said is in plasma and extracellular fluid, or it may be recognized by pathogen recognition receptors that are in macrophages, which are in many tissues, are resident macrophages are in many tissues, and also by neutrophils that are in the blood, for example. If the extracellular pathogen is in contact with epithelial cells, we talk how they form a barrier that prevents infection. And one of the barriers are the chemical barrier that include the production of antimicrobial peptide that will promote the destruction of the pathogen, because one way they do it is by damaging the membrane, forming a pore in these bacteria, for example, the same as the complement can form a pore as well. And so if the pathogen is intracellular, so it grows inside of the cell, it may grow in the cytosol, viruses, for example, and in this case, we learn that natural killer cells can kill these virally infected cells, or it may grow in the vesicular compartments. So, for example, many protozoan parasites or mycobacteria are able to grow in the endosome of the macrophages. But the macrophages, when may uptake these parasites, and when activated, many times they are able to degrade and promote the degradation of these parasites. Many times, for that to happen, they require further activation by the adaptive immune cells, and we will learn that later. And so we also learn how extracellular microbes can be recognized by pathogen recognition receptors. And obviously the pathogen recognition receptors that are localized in the surface of the cell, in the plasma membrane, are more suitable to see, right, to recognize and bind the extracellular microbes. So toll-like receptor 4 and toll-like receptor 5 can sense extracellular microbes. Although this is not absolute, this is in generality, and the cytosolic pathogen recognition receptors like RIG-I or SIGAs or the node-like receptors, they are more suitable to sense the intracellular pathogens. And then we learned that some of the pathogen recognition receptors, like toll-like receptor 3, 7, and 9, are localized in the endosome, and they will recognize vesicular intracellular pathogens. Okay, so we will leave this question as well for the discussion section. And so just here to summarize, we talked today about the recognition of the microbes by cell-associated receptors that are localized in the surface and the cytosol. We talk about the toll-like receptors, the card sensory proteins, the node-like receptors, the SIGAS enzyme, and how they all act through an adapter and signaling via kinases, activating transcription factors that induce the production of cytokines. And these cytokines will alarm, right? And we talk about 4 ways in which cytokines will alarm. Number 1, by producing the synthesis of mannose binding protein and C-reacting protein by liver, activating other cells, and we talk how interferons can create an antiviral state in a paracrine manner. They can also activate natural killer cells. Number 3, the cytokines may recruit other cells, and we talk how cytokines form a gradient to recruit cells to the site of inflammation, like neutrophils, for example, that are in the blood can be recruited. Number 4, the cytokines might have systemic effects that may include, for example, the induction of fever. And then we talk at mechanisms of destroying the microbe, and we mention again phagocytosis by macrophages and neutrophils, cytotoxicity, and we talk about how natural killer cells can sense the presence of a virally infected cell that downregulated the expression of MHC class 1 and can kill the viral infected cells. And also we have mentioned in past lectures and today that complement and antimicrobial peptide may also promote destruction of the pathogen by damaging the membrane of the pathogen by creating a pore. And so with that, this is in a nutshell really what we want to cover about the innate immune system. And so in the next lecture, we will start with more about the adaptive immune system. And so as always, I'm happy to stay if you have questions. And I also have office hours starting in 10 minutes. Thank you. Any question?
I had a question about how you mentioned that LPS is a PAMP at the beginning of the lecture.
Yes.
Now, I was wondering, like, is that like I'm guessing like not all gram-negative bacteria are pathogens. Is that right?
That's true, because they can be commensals and you not necessarily classify them as pathogens.
So like how do the innate immune cells like distinguish between like the good gram-negative bacteria and the bad ones if they all have, if LPS is a PAMP?
Yeah, that's an excellent question. Actually, that's a question that I think that has been long asked in the field. And there are different type of explanations. One is that not all the commensals will be in contact with macrophages because they are in the lumen of the intestine, for example. Then after that, they only are in contact if they cross the mucus layer and the epithelial cells, for example. And so not necessarily are in contact with the PRR, okay? But some may be, and some dendritic cells have been also shown to have prolongations of their cytosol that cross into the lumen. So, and they can have expression of TLR4 dendritic cells. So you're right that the commensals may also activate TLRs. And what happened is that this seems to be important to induce some type of inflammation or that will keep in check these commensals. And actually, if you lack MYD88, for example, there may be an outgrowth of the commensals, and that may cause a colitis. Or other type of inflammatory gut diseases. And so the innate immune system can recognize these commensals and induce a mild inflammation that keeps them in check. So now, normally these commensals are not virulent, and so they typically will not infect the cells and replicate inside our cells. And so they may activate toll-like receptor 4 because it's in the surface, but they will not activate node-like receptor, for example, that is necessary for the activation, further inflammation and activation of caspase-1 that induce the conversion of pro-IL-1 beta into IL-1 beta, and it's another layer of more of inflammation that now may induce systemic fever, right? So that explains why we don't, not all the, it's a mild inflammation that keeps the commensals in check, but it's not to the degree that is induced by a bacteria that may be pathogenic and will invade the cell and may replicate in the cytosol of the cell, and therefore will activate other pathogen recognition receptors, not just the ones in the surface, that will escalate the inflammation to contain now this bacteria that is more dangerous.
Okay. Okay, thank you.
Sure. So I have office hours in 4 minutes. So if there is no pressing question, so I will take these 4 minutes for a bio break and then come back at 2.
Thank you, Professor.
Okay, thank you. See you in a minute.